Well, hey, AP Bio, hope you're doing well when you are watching this. This is lecture 5. We're going to be covering 3 topics on AP Classroom when we do this lecture. It's going to be multiple topics, 7.6, 7.7, and 7.8. And so 7.6, again, changing genetic makeup, right? Changing alleles. And so describe the types of data that provide evidence for evolution. Well, I already talked about this, so we're going to kind of run through this again. But a lot of different evidences, like evolution is supported by scientific evidence from many disciplines: geographical, geological, which like fossils, physical, biochemical, mathematical even. Fossils, we're going to briefly dive into fossils. Not going to dive into that crazy amount. But fossils, I'll briefly talk to you about how like carbon-14 dating happens. Again, homologies, morphological, which is like vestigial structures, and it's shared by common ancestry, which is something Darwin thought about, and a comparison of DNA sequences, which we'll practice in class and also in this video. And then we're linked by lines of descent from common ancestry. So describe the fundamental and molecular and cellular features shared across all domains of life. And I'll talk to you about that. Many fundamental molecular are conserved. Conserved is a key word in evolution, which means they're so necessary for survival that they have not changed, right? And then structural and functional evidence supports the relatedness of all organisms in all domains. So that's 7.6. 7.7 is common ancestry, which I kind of briefly just talked about. But some specific examples, again, we all came from the same place. And so describe structural and functional evidence on cellular and molecular levels that provides evidence. Well, membrane-bound organelles, we're going to talk about endosymbiotic theory again. Eukaryotes, that's us, common ancestry, linear chromosomes, and genes that contain introns, which means, introns are the ones we don't want in the final RNA transcript. And then finally, 7.8, continuing evolution. Life continues to evolve. Evolution just hasn't stopped. We are definitely continuing to evolve. Life is, and so we'll look at how genomic changes. Hold on, let me see if I can do this. Genomic changes over time, continuous change in the fossil record. We see a lot of different stuff there. A resistance to antibiotics and pesticides and herbicides and even chemotherapy drugs. Like that is evolution. Pathogens and how they change and evolve. I mean, of course, living post-COVID, but COVID changed a lot. Bacteria changed a lot in response to antibiotics. So we'll talk about all that. So we're going to kind of run through this. Again, I'm going to review a lot that we've already talked about a little bit, add a few things, and practice some questions with you. So here's good old Darwin. After his voyage on the HMS Beagle, there's the Beagle. 5 years, again, where a lot of this stuff took place, where his thoughts were really cemented was the Galapagos Islands. And there's Galapagos. And there's the HMS Beagle. 5 years. Can you imagine it? So Darwin perceived adaptation to the environment and the origin of new species as closely related. You have to be able to adapt, which is new traits that help, you be fit. And so, adaptations include, and he documented this, Darwin did, finch beaks. And so you have the cactus eater, which is the long, sharp beak. Helps it tear and eat up flowers and pulp. Then you have the seed eater, which has a large beak adapted for cracking. Adapted, right? Key word, adapted. Adapted for cracking seeds. Well, how did this happen? They choose just to have big beaks? No. It's just, according to evolution, random mutations and differential survival rates based upon these adaptations. Uses, an insect eater, narrow beak, to grasp insects. Okay? And so Darwin wrote an essay about natural selection as the mechanism of descent with modification. So we're descended from common ancestor, yet we've accumulated changes or adaptations. Natural selection is a process by which individuals with favorable, never hurts to review, favorable inherited traits are more likely to survive and reproduce. Again, can't draw a line. That is the goal in biology, okay? A guy by the name of Wallace also was kind of thinking about this theory at the same time. Darwin wrote his book when he figured out he was kind of working on the same theory. Darwin reached it first, but Wallace also reached a similar understanding. And so because of that, it's often, he's often given credit for that. There's a great video. I highly doubt you have time to watch it, but just, making a theory. Okay, so Evolution by natural selection is a blend of chance and sorting, which means like it's just random chance. But again, natural selection does favor some alleles over others based upon the environment and based upon survival of the fittest. Okay, and so with that being said, there's different traits, right? Qualitative traits influenced by alleles at one locus. Quantitative traits usually influenced by more than one allele. And so you can kind of read this: struggle for existence and struggle for survival of the survival of the fittest. Not everything is directly competing, though. A moth may have more offspring than other moths because bicolor is more effectively concealed from predators. It's not directly competing with another moth, but it still has an advantage. So certain traits lead to fitness. Although we often refer to the relative fitness of a genotype, remember that the entity that is subjected to natural is a whole organism, right? Not just that one genotype. So selection acts more on phenotype than that. And so there's a couple of things you have to have: variation, inheritance. You have to have variation. You have to have differences based upon genetic differences. So many differences are heritable. You have to be able to pass these differences on. Differential adaptiveness. Some differences affect survivability and differential reproductive. Some differences affect the likelihood of surviving and reproducing. So natural selection results in a change in allele frequencies of the gene pool, improved fitness of the population. There's a couple of different ways natural selection can act on quantitative traits: stabilizing, directional, and disruptive. Stabilizing is often called purifying selection, and we're selecting for the mean. Okay, an example of this is birth weight. You can see that low birth weight and high birth weight increase mortality, and for a lot of different reasons. So this would be like a stabilizing selection. And then you can see as birth weight when you're low and when you're really high, there's a lot of respiratory distress when you're like underweight. Okay. That's just one example. When you're overweight, you can also have issues. Positive selection is usually in a certain direction. So example is this like bush here with spines, right? And so warthog's trying to eat this bush, but more spines would mean less chance of being eaten and destroyed, and that trait would pass on to future generations. So that would be a positive, a directional. Much like longhorn cattle, these horns have been selected to be longer because it helps protect and ward off predators from the calf. And then disruptives, like the weird one, but we favor the extremes. Okay, and so this is obviously highly seen in specific environments, but one example is the black-bellied seed cracker, bill sizes. As you can see right here, there's a lot of, on this, in their environment, there's a lot of small seeds and a lot of large seeds, but not many in the middle. And so birds with smaller seeds, smaller bills, can feed efficiently on soft seeds, small seeds. Large-billed birds can crack the hard seeds, but birds within the middle, they don't really succeed at both, and so they can't really survive as well as the other birds. Natural selection. There's something in natural selection called sexual selection. And these are adaptive changes in males and females lead to an increased ability to secure a mate. Males compete, females choose. There's a couple of hypotheses about that. Don't really need to know that too much for our purposes right now. But dimorphism, if you see a pretty bird, usually that bird is a male because of sexual selection. Here's, drab female, peacock's the male, right? Male and female common pheasants, male and female lion. And so males compete for territory, for mates, and the strongest wins. And so in theory, these good genes, if you look beautiful, if you look healthy, if you compete and win, you have good genes to pass on. So that's like a hypothesis. There is selection among humans as well. In not all cultures. Some cultures arrange marriages and some belief systems, but yeah, and we could talk all day long about being in high school and promposals and trying to dress a certain way to be attractive, but we don't have time because we're in AP biology. So moving on. Darwin viewed the history of life like a tree with a common trunk representing shared ancestry. And fossils of extinct species help to fill in the morphological gaps between present-day groups. There's a lot of assumptions with that. Don't have time to dive into it. But this is what he drew in his book. So we have a common ancestor. And then like you have other related species over here, and related species over here, and related species over here. And so it looks very similar to, here's an example. I'm sure I've shown you this before, but elephants, modern day living elephants. There's a common ancestor right there, right? And we're talking, crazy amount of time ago. And millions of years ago, you can have, a way far common ancestor. And so this would be like the simple modification. They're all related. This would be like an outlier. Yes, a manatee is closely related to an elephant compared to other organisms. But, these are still kind of outlier groups. And then you can have common ancestor all the way down. Okay? The endosymbiotic theory, you have the printout in your packet. We talked about this before, but this is an example of, of evolution. And how eukaryotes share common threads, So prokaryote, in first, ancestral eukaryote consumed aerobic bacteria, okay, mitochondria, right? And then there's like this common ancestor that diverge in photosynthetic. Okay, we're not going to destroy this, so modern photosynthetic eukaryote, modern heterotrophic eukaryote. And then here are our evidences that y'all are done so well. Remember, Mad Doctor. Evidence supporting common ancestors for all eukaryotes is membrane-bound organelles. Let's talk about that, endosymbiotic theory. Linear chromosomes, not circular like we see in prokaryotes. Genes that contain introns, which again, you want introns out. You don't want introns in. You want introns not in that final transcript. So you want them out of the final mRNA. Linear chromosomes, here's an example. Eukaryotes contain multiple linear chromosomes. So again, this is something we all have. Captive telomeres found inside the nucleus. Genomes are larger. Prokaryotic chromosomes, circular, occurs in the cytoplasm. Genomes are smaller. Okay, so that again, that's example of evolution in eukaryotes. Okay, so let's go through these questions together. And I would pause it before I tell you the answer. So I'll tell you when I'm telling you the answer. But researchers observed selected internal structure of 4 different microscopic organisms as part of a larger study on the divergence between eukaryotes and prokaryotes. The observations are recorded in this figure. You'll see a lot of figures on the test. Which organism would the researchers most likely predict to be the most distantly related to eukaryotes? You have 1, 2, 3, 4. Here's your key. Go ahead and pause it. Think about it. I'll tell you the answer. All right, so the answer is A. Circular chromosomes. That's a prokaryote. Moving on. Here's a spliceosome. Moving this removed intron right here, exon 1, exon 2. We keep the exons. Which of the following best predicts the importance of this process? For time in making the video, go ahead and pause. Think about the answers. All right, hopefully you answered it. You gotta think about these, so don't just rush to the answer. But the answer is A. Let me see if I can write on this. The answer is A. Nice. All right, not very good drawing, but nonetheless. And then finally, this one's a little bit more challenging, so I'm going to kind of walk you through this one a little bit. Transposable elements are significant features in the genomes of almost all eukaryotic organisms. The accumulation of these elements in various species can help determine their relatedness. Table 1, you'll see tables like this on the test, on my test and on the test in May. Table 1 indicates the presence or absence of 5 segments of DNA that have been moved by transposons, in 6 primate species. Okay, so to kind of tell you a little bit, transposable elements are in the genomes of almost all. Okay, these are just elements, not too much more. Which of the following best predicts how phylogenetic relationships might be revised if transposon 1 was not found in chimpanzees? Okay, so what they're asking for is this is evolutionary relationship. It's interesting that they asked this in the topic that we're in without covering phylogeny. But phylogeny is basically common descent, like a tree of life. Okay, so how would basically evolutionary history, common ancestry, be affected if transposon 1 right here was not found in chimpanzees? Okay, and so right here, hopefully you can see that these 3, let me do a different shape. These 3 guys are are like share a lot of similar traits, right? Like they share a recent common ancestor, and now these two guys also share a recent common ancestor because they still have a presence of one of the DNA sequences. But if this is now a negative, it's no longer, the chimpanzee would no longer be considered closely related to the bonobo. I probably butchered that, but it would be more closely related to the human being. And so the answer is B, because it now would not have that common line of DNA, that commonality, right? It would have it now with the human, because the human shares the most recent common ancestor. Direct observation is going to fly through this because I talked about it before. And, you know, again, probably shouldn't have, but can't hurt to review. So, biology is evolutionary change. You know, we talked about the soapberry bugs in Florida in a previous video about how, you know, there's their environment. Southern Florida has, you know, larger seeds and fruits. Central Florida has smaller fruits, and so, you know, they've adjusted their beak length, not on purpose, just natural selection. We talked about superbugs and MRSA and how they can evade bacteria and pathogens. You know, 2 years after penicillin was introduced, Staphylococcus aureus became resistant. Again, that's just random because bacteria replicates so fast. It's just random change. Natural selection does not create new traits, but selects for traits already. What does create new traits? We talked about this before, but that is mutations. Mutations select for new traits that create new traits. Okay, so evolution can occur rapidly when organisms reproduce rapidly, because you get a lot of changes. So viruses and bacteria and all sorts of stuff. Homology is similarity resulting from common ancestry. So, for example, homologous structures are something like this. Homologous structures are anatomical resemblances. So you can see right here you have one bone, couple bones, many bones, and you can see all across these vastly different, well, you know, relatively vastly different species. You can see similarities in structure. Comparative embryology reveals anatomical homology is not visible in adults. So, for example, we have pharyngeal arches as human beings, postanal tail as human beings. We must have evolved from a similar place according to evolutionary theory. Vestigial structures are remnants. For time, I'm not going to. Well, actually, let's just see if it works. I don't know if this is going to work recording or not, but let's just see. Maybe. This is kind of interesting. I was trying to find this. I mean, everybody goes to like wisdom teeth and, you know, various things like the appendix, right? Legs in a snake, whale pelvis, all that sort of stuff. But there's this really interesting website, the pink part of your eye that's actually useless. That pink corner of your eye, right next to the bridge of your nose, is kind of a feature that you look at all the time without really noticing, but it's actually a fascinating relic of our species' ancient ancestry. Small fold of conjunctiva, which is like the covering of your eye, the mucous membrane that covers the whites of your eye. In humans, the plica semilunaris appears to do nothing. It's the last surviving remnant of a third eyelid called a nic. Sorry, something froze. But yeah, nictitating membrane, which lies beneath the outside. I mean, there's that third eyelid kind of going sideways. I mean, it's interesting, right? And you could have some interesting discussions when it comes to faith and all of that. Does it have any uses now? You know, who knows? Just something interesting that I kind of found. Molecular homologies are shared genes. So homologous genes can be found in all organisms that are dissimilar. So a famous one for this is cytochrome C, which is an electron transport chain in mitochondria. And so you can see that they've built a related phylogenetic tree, and it's from the Encyclopedia Britannica. Super exciting there. And so, but cytochrome C, again, protein in the ETC. And you can see that we don't have any differences between monkeys and humans, about one or so. We have, you know, accumulated 7+ differences. You know, this is just kind of how many differences exist. You just kind of keep adding and adding and adding. So we have a lot of differences when we start moving this way. So our differences increase as we move this way. And so that means we're less related to these guys and these guys, which, and again, that's just based upon genetic amino acid, like nucleotide differences in cytochrome C. So for example, this is just an example from another textbook I have, but cytochrome C contains about 100 to 104 amino acids, which means, 300 or so, give or take, nucleotides, codons, right? This chart shows how many differences exist between two species. Man and monkey has 1 difference. Man and dog has 13, 18, right? Various, so those differences, according to evolution, accumulated over time. And so that is an example with that. A comparison of DNA nucleotide sequences provides evidence for evolution. Same or highly similar genetic code means we are more commonly we share a more common recent ancestor. So for example, you can see that humans share about 8.8 of mitochondrial DNA sequences. 8.8%. Gorillas, orangutans, gibbons. And so, based upon DNA, we're more closely related to the chimp than we are to a gorilla. Convergent evolution or analogous features in distantly related groups because it's similar traits, but that we're not sharing a common ancestor, a recent common ancestor. So we talked about like the flying squirrel and the sugar glider. Fossil record is, again, a lot of people like to see this as Exhibit A for evolution. It documents a pattern of evolution. And so, we can see how over millions and millions of years, according to evolutionary theory, how ankle bones have come to be. Fossils can document important steps of transition, And so here's a common ancestor. There's Pakicetus. Again, whale evolution. It's very interesting. Life began in the sea. Mammals went to land, went back to the sea. If you dive into it, there's a lot of questions that come up. But, this is just something to know, that this is evolutionary theory. As far as fossils go, fossil evidence shows that living cetaceans and their close relatives are more different from each other today than were early cetaceans and even toed ungulates. So sometimes you'll hear these finds like, they found a rock that was X amount of years old. Well, they do that with radiometric dating, and so it's used to date materials such as rocks using radioactive materials. It's been a long time since I've talked about this stuff, but basically there's an assumption here about like how many, carbon-14 atoms were there at the beginning. And then I'm not going to talk in ignorance here, but basically a half-life is 5,730 years. So what they do is basically you start with a number of carbon-14 nuclei. After 5,730 years, you would have half of it remaining. And then after another, you would have a fourth left. And so this is just a way to kind of measure rocks around, fossils and, just radioactive decay. Biogeography is another evidence of evolution, the scientific study of geographic distribution of species. So the example is marsupials. These guys are only found in Australia. They're isolated to Australia, while placental mammals dominate other continents. Marsupials are isolated to Australia. Why does this matter? It supports continental drift, Pangea, and it shows how species evolve separately when geographically isolated. Koala, wombat, kangaroo. Islands generally have many endemic species. Just talk about Australia, which means these guys are nowhere else to be found on the planet. Talk about ecotourism and all that. And so it's just interesting to think about. So you can see right here, resistance to pesticide. This was some data that I don't know if I gave you or not the first go around, but this was a commonly used pesticide, 70s or so. You can see after time, their resistance went up, right? And why is this? Because the mosquitoes that had a variation, right, that led them to survive when DDT was applied, survived and passed that trait on, which natural selection selected for. That's probably should use a different word than selected for, but that's an example of that. Okay, and so you can see that's an example. So let's go through some more examples again. Again, try to answer it before I give you the answers. Students analyze several photosynthetic eukaryotic organisms in the laboratory to determine their phylogenetic relationships. Again, haven't discussed crazy amounts about that, but basically evolutionary relationships. Organisms were Spirogyra, a green algae, a moss, a spruce tree, and an apple tree. Table 1, here's a table. So again, you're looking for similarities. When you see a table like this, and we'll practice this a lot more, but you're trying to find similarities, right? So this is an outlier. There's nothing else that shares it. There's 3 guys that share this, 4 guys that share this, and then the moss is kind of an outlier there as well. Okay? And so, which of the following rows of data listed in Table 1 best supports the possibility of a common ancestor for all the organisms listed there? Okay? And so what you're thinking about is, well, common ancestor for all of them, well, that trait has to be present in all of them. And so go ahead and think about your answer. Pause. And the answer, I wonder if I can do something cool here. You're like, bro, this is long enough. Like, don't. But yeah, it's, oh no, where's the, oh, you can't see it because it's like so, like, crazy. Let's do something else. You can't see. Oh, I needed like a yellow. But it's C. That's better. Such a waste of time. Cell wall composed of cellulose. Why? Because hopefully you can see that that was the wrong thing. But they all have the positives. They all have that, the cell wall composed of cellulose. They all have it. They all have that trait. And so you can't have a common ancestor if you don't share all the traits. Here's another example of a table. Researchers studying the evolutionary relationships of 3 species of snails collected the data shown in Table 1. All right, species A, B, and C. Location of fossils, shell orientation, shell composition, trophic level. I haven't talked about that, but they're all carnivores. The trivores basically eat like detritus, which is broken up stuff. And DNA analysis, which is how much, their A's and T's and G's and C's. You're like, shell orientation. When we talked about snails, well, this is an example of just seeing, right, an example of some new data that's falling within the lines of what we've been talking about, these huge big picture items. So which of the following data sets is most likely to provide reliable information about the evolutionary relationship among these 3 snail species? So out of all of that data, which one would you think shows the best or shows the best information about evolution? Pause. All right. And the answer is, drumroll please, C. Hopefully fossils was like, okay, yeah, I'm going to pick fossils for sure. Shell orientation's like interesting, but that is based upon DNA, developmental DNA. Shell composition is kind of what's in the environment. Trophic what's in the environment. DNA analysis has nothing to do with any sort of gene relatedness. So that's just an example there. Another one. Hemoglobin is a highly conserved, which means hasn't changed very much through history because it's necessary for this common lineage. Protein used by all mammals to transport oxygen. Table 1 was constructed by comparing the amino acid sequence in the hemoglobin molecules from 5 different mammal species. All right, so you got to think about what they're giving you. What are they giving me? Hemoglobin amino acid differences among 5 different mammal species. So we know they're related. I always like to circle low numbers right from the get-go. Okay, 3's there, 5's there. 10, 11 is kind of similar, right? Which of the following best describes the importance of the number of amino acid differences indicated in Table 1? Why are we even looking at this is the question. Okay, pause. Go ahead and answer. And the answer is B. They reflect the number of base pair changes in DNA. You're like, well, I thought it said amino acid. Well, 3 nucleotides encode for amino acid, right? So change in base pairs in DNA is important. And then finally, the last topic, which was continuing evolution. Genomic changes over time lead to all species evolution. So changes in DNA, like gene mutations, right, chromosome mutations, that's a foundational, we have to have those in order to have evolution. Again, mutations are mostly neutral, then mostly negative, and then some of them are positive. So some questions that hopefully that brings up to you. Cell division is sexual reproduction, increases gamete diversity. So you have fertilization, independent assortment, which is metaphase 1, crossing over, which is prophase 1, and then environmental disruptions. The environment is changing. In ecology, we'll talk about global warming. And so these will put different pressures on populations and cause them to change, maybe even rapidly, or maybe lead to extinction possibly. Continuous change in the fossil record is evidence. Fossils are a preservation or an impression of an organism that lived in the past. Provides a record of an organism over time. Strata layers often arrange fossils in the order that they were deposited. There's some interesting data that shows that's not always the case. If you want to look on, I can talk to you about that in class. New fossils mark changes in the environment. So new selective pressures, right? Different, maybe like climate change, right? Dramatic. The Bible teaches about a worldwide flood, and evolution teaches about I guess the asteroid hit the Yucatan Peninsula, and stuff. Although, there's matters your worldview lens, right? But fossils, rapid extinction, pretty quick. Resistance to various chemicals has resulted in a species evolution. We talked about this when we talked about the mosquitoes, right? So, for example, you spray a pesticide. These guys are resistant just because they have some sort of enzyme that allows them to not die when the pesticide's sprayed. And then if you keep using that pesticide, well, all it does is increase the likelihood that these guys survive and reproduce. Pathogens is something that causes suffering. Pathos means suffering. Gen means beginning of. And so these guys, pathogens, evolve and cause emergent diseases, bacteria, and viruses, and all that. Some great examples is these pandemics that we've had. The swine flu, which, I don't even know if y'all were alive then. Were y'all alive then? I guess you were. Juniors and seniors. But a new strain of H1N1, caused by reassortment of the genes. So these genes transpose, different properties came to be. The H protein was originally only seen in pigs, the one that attacked humans. COVID-19, of course, there's been 15 different variants. Even more than that. All right, so let's go through these questions. We got 3 more questions, and then we'll call it a day. Researchers examine the ability of cultures of the bacterium Pseudomonas aeruginosa and to adapt to the antibiotics ceftazidime and avibactam. When they were minister, I should have checked out how to say that before making the video. The researchers grew 4 replicate cultures in growth medium with no antibiotics added. Okay, that's control, right? No antibiotics. And 4 additional replicate cultures in the same growth medium with added ceftazidime and avibactam. I don't know. At the conclusion of the experiment, samples of the replicates exposed to antibiotics were removed and their genomes were sequenced. Alright, so basically you've grown some bacteria in a control and then other bacteria are exposed to a selecting agent, an antibiotic. All of the antibiotic-resistant mutants were missing 3 genes. Alright, so here's the data. Right? Missing these 3 genes. Antibiotic resistant. I would underline that. It is known that one of these genes makes wild type susceptible to antibiotics, right? However, the researchers do not know which gene it is. Okay, there's a problem. Which of the following experiments would specifically determine which gene is responsible for ceftazidime avibactam sensitivity in P. aeruginosa? Wow, that was insanely hard to read. I should have proofread that. All right, we'll go ahead and pause it. So again, remember, selecting agent is this antibiotic. One plate doesn't have it, other plates do. But we're trying to figure out. We know one of them. One of these 3 genes has to, cause this bacterium to be susceptible to the antibiotics, right? But how can we figure out which gene it is? And the answer, hopefully you use just process elimination here. Haven't truly talked about gene knockout yet this year, but it's this one. We gotta basically knock out this gene to see and create mutants. And we should create one line that has each gene knocked out and see which one survives or dies when the selective pressure is added. Low doses of antibiotic are often added to livestock feed to increase production. Studies have shown that bacterial populations constantly exposed to the sublethal dose can evolve resistance. Research group claimed that when a population of bacteria are constantly exposed to a sublethal dose of streptomycin, their fitness declines due to the increased energy requirements for survival in the presence of the antibiotic. For subsequent studies, the researchers wished to determine whether adding a low sublethal dose of an additional antibiotic causes further decline in the fitness of the bacteria. So what's the most important? Again, always underline stuff, right? What's the most important things? Right here, they claimed constantly exposed to sublethal doses of streptomycin. Claim that. Researchers wish to determine whether adding another low subdose further decline the fitness. Which of the following best represents the next step? This is like basically scientific design, right? And so go ahead and pause. And hopefully, we're thinking low doses, right? So anything that's at high dose would be bad. No dose wouldn't work, so the answer is B. And then finally, the last one. This is a fun one, very long. Staphylococcus aureus is a pathogenic bacterium that can infect a wide range of host species, including humans. It has a particular protein that binds with hemoglobin for the host organism. Hemoglobin is in red blood cells, right? Hemoglobin is an iron-containing protein used to transport oxygen in the blood. Since iron is important for growth, Staphylococcus aureus have evolved the ability to absorb the iron from the host's hemoglobin. Tricky little guys. Different Staphylococcus aureus strains preferentially infect different hosts and have different amino acid sequences as their hemoglobin binding domains, which that makes sense, right? Because hemoglobin differs in different organisms. In experiment, different Staphylococcus aureus strains were mixed with hemoglobin from macaque monkeys, and their binding ability was measured. All right, here we go. So here's some data, right? We got strains 1, 2, 3, and 4. Remember, amino acids all have different letters. You don't even know which letter means what, but each letter stands for a different amino acid, which means, again, different DNA, right? And then species A, species B, macaque, and species C. Oh man, I didn't get the rest of the hemoglobin binding thing. Hold on. Alright, sorry about that. I had to go find this graph here, which is super important. Alright, so you can see right here the data represent hemoglobin binding. Alright, strain 1, 2, 3, 4. Which of the following experiments would be most appropriate to determine whether populations of Staphylococcus aureus are continuously adapting in order to obtain iron from hosts more effectively? Alright, I will go ahead and pause it. Try to figure it out. And the answer is hopefully you saw the key word like many generations here. Evolution over time, right? And so this is D. Like I said, that we are wanting you to be able to think. So again, being able to think is important. So hey, long video, but I hope it was beneficial. Hope you learned something. We'll talk and practice more in class. Much love in Christ, guys. See you.